Hello, everyone. Welcome to this teaching and learning video series for Semester 2. In this lesson, we'll focus on oxidative phosphorylation, the final stage of aerobic respiration. This is where our cells generate energy in the form of ATP, which powers almost every biological process. It's an essential step that uses oxygen to help produce energy efficiently inside the mitochondrion. In the next part, let's explore where this process occurs and how it produces ATP for the cell. Now, let's look at oxidative phosphorylation in more detail. This is the final stage of cellular respiration, and it takes place in the inner membrane of the mitochondrion, also known as the cristae. This stage is extremely important because it produces almost 90% of the ATP used by the cell. Here, the energy that was carried from earlier stages of respiration, NADH and FADH2, is finally converted into ATP. Oxidative phosphorylation consists of two main stages, the electron transport chain and chemiosmosis. Now, let's explore the electron transport chain. In this stage, several electron carrier molecules in the inner membrane of the mitochondrion pass electrons from NADH and FADH2 through a chain of redox reactions that release energy. Before we look at how the electron transport chain works, let's first identify its main components. There are four main protein complexes located in the inner membrane of the mitochondrion. Complex 1 is called NADH dehydrogenase. Complex 2 is succinate dehydrogenase. Complex 3 is cytochrome C reductase, and complex 4 is cytochrome C oxidase. In addition to these complexes, there are two mobile electron carriers, which are coenzyme Q, also known as ubiquinone, and cytochrome C. These carriers help transfer electrons between the complexes. In electron transport chain, the oxygen acts as the final electron acceptor at the end of the chain, combining with hydrogen ions to form water. Together, these components drive the flow of electrons and the release of energy needed to make ATP. Now, let's see how the electron transport chain works. First, NADH transfers its electrons to NADH dehydrogenase, also known as complex 1, and becomes oxidized to NAD+. Next, FADH2 transfers its electrons to succinate dehydrogenase, or complex 2, and is oxidized to FAD. The electrons released from both NADH and FADH2 are then passed to coenzyme Q, which will carry them to the next complex in the chain. Then, coenzyme Q passes the electron to the next carriers, which are cytochrome C reductase, cytochrome C, and cytochrome C oxidase. These transfers occur through a series of redox reactions that continue to release energy. Finally, the electrons are transferred to oxygen, which acts as the final electron acceptor in aerobic respiration. Oxygen then combines with two electrons and two hydrogen ions to form a water molecule. This step completes the electron transport chain. Next, let's look at chemiosmosis, which is an energy coupling process that links the movement of protons to the production of ATP. During this stage, protons that have been pumped into the intermembrane space flow back into the mitochondrial matrix through an enzyme called ATP synthase. As the protons move through ATP synthase, the energy from their movement drives the conversion of ADP, an inorganic phosphate, into ATP. This is how chemiosmosis produces the cell's main energy molecule. Now, let's see how chemiosmosis actually works. When electrons move along the electron transport chain, they release energy at each step. This energy is then used to pump hydrogen ions, or protons, from the mitochondrial matrix into the intermembrane space. Three main protein complexes act as proton pumps. These are NADH dehydrogenase, cytochrome C reductase, and cytochrome C oxidase. As the protons accumulate in the intermembrane space, a proton gradient is formed, creating a strong proton gradient and generating what we call a proton motive force. These protons then flow back into the mitochondrial matrix through an enzyme known as ATP synthase. This movement occurs down the concentration gradient. The flow of protons provides the energy needed for ATP synthase to catalyze the formation of ATP from ADP and inorganic phosphate. This is the final step of oxidative phosphorylation, where the cell's main source of energy, ATP, is produced. Now that we have seen how oxidative phosphorylation works, let's look at how much ATP is produced from the electrons carried by NADH and FADH2. During glycolysis, two NADH molecules are produced. When these NADH pass their electrons to oxidative phosphorylation, they generate 6 ATP. From pyruvate oxidation, another two NADH molecules transfer their electrons, producing another 6 ATP. In the Krebs cycle, 6 NADH and 2 FADH2 are formed. When their electrons enter oxidative phosphorylation, the 6 NADH produce 18 ATP, and the 2 FADH2 produce 4 ATP. Altogether, oxidative phosphorylation produces about 34 ATP, which represents the main energy output of aerobic respiration. Let's summarize the total ATP produced from the complete oxidation of one molecule of glucose. From substrate-level phosphorylation, 2 ATP are formed during glycolysis and another 2 ATP during the Krebs cycle. From oxidative phosphorylation, NADH and FADH2 donate their electrons to the electron transport chain to generate ATP. The NADH from glycolysis produces 6 ATP. The NADH from pyruvate oxidation produces another 6 ATP, and the NADH and FADH2 from the Krebs cycle produce 18 and 4 ATP respectively. When we combine ATP from both substrate level and oxidative phosphorylation, the complete oxidation of one glucose molecule yields a total of 38 ATP. This shows how efficiently our cells convert energy from food into usable ATP. And that brings us to the end of our lesson on oxidative phosphorylation. Through this process, we have seen how electrons from NADH and FADH2 move through the electron transport chain, creating a proton gradient that drives the production of ATP. This amazing mechanism shows how our cells efficiently convert the energy stored in glucose into usable energy for life processes. Thank you for watching, and I hope you now have a clearer understanding of how aerobic respiration produces energy for every living cell.